of mutations in your somatic cells, but if it's not occurring in your germline cells, it's not going to be passed on to your kids. So it might be a major bummer for you, but it's not going to affect your kids, which means it's not going to get passed on to the population, right? Okay. Soaking it in. Right, so that's it. That's the answer. Somatic mutations might affect the individual, but they're not going to affect the population, right? And what we care about are genes becoming a part of like the human genome. They have to be passed down through our germline DNA. So it's right. So if you have low rates of mutation in a human, that means it's going to take kind of a long time for changes to occur within the genome of the population. But low rates of mutation in single-celled organisms like bacteria can yield high rates of evolution in the population. There's two reasons why. Does anyone want to take a guess at why? So I'm saying in us, we have low rates of mutation and it takes a long time for that to affect the population, but that's not the case in bacteria. Yeah?
So they reproduce so quickly.
Oh yeah, they reproduce like gangbusters. You have one E. coli right now, put it in a culture tube, you will have millions by tomorrow, right? You have one me. Over the course of my entire lifetime, I have had 2 children. I'm not having any more because we cannot be outnumbered. And I will not survive. We understand our limits. I can't even handle a cat right now. We're good. So right, they reproduce really quickly. There's one other reason, and it kind of has to do with what I've written here. Yeah.
Not only can they reproduce vertically, they can have horizontal gene transfers. They don't have to reproduce to share their genes.
Snap. That wasn't even what I was thinking of, but that's a really great observation. Yeah. So they have other forms of ways of transmitting their genes. Yeah. Yeah, right here.
How about if the mutation that occurs leads to the survival of that, so then the further population will all have that mutation?
Oh, that's really, gosh, you guys are coming up with things that I wasn't even thinking about. That's great. Yeah, for sure. Right, we see this with antibiotic resistance, which can occur really quickly and kind of lead to even increased survival of the bacteria, and so you get even more increased proliferation. What about, do mutations have to affect bacteria germ cells? Do bacteria have germ cells? Yeah, they're just one cell type, right? For us, we have trillions of cells, and I'm saying that the mutation has to occur specifically in our germline. Bacteria, if they acquire a mutation, it is getting passed on, right? Because it's just themselves, and they divide and they pass on their entire genome, right? Where we only pass on our germline genome. Does that make sense? Okay. They do not have sperm or eggs, it is just them. Okay, right, so when I say, right, this is touching on the question that was asked over here about, mutation is rare. How rare is it, right? So there's an average mistake of 1 base pair per 10,000 base pairs that are replicated. That's kind of a lot if we have 3 billion base pairs. Thankfully, we have all these sweet proofreading mechanisms that I think you learned about back in Bio 2, and maybe genetics too. So proofreading and repair mechanisms do a pretty good job of cleaning up the mistakes that occur, and it declines to 1 per billion base pairs. So that's not too bad, right? And there's a 1 in 3 chance of it hitting the wobble position. And then, right, if only 8% of our genome is either genetic regulatory elements or genes, odds of it making a really meaningful change are pretty low, right? So that's what I mean by low rates of evolution during a single replication event. Does that make sense? Ish? Okay. I feel like it's mean to have classes at 3 p.m. Is everyone tired at 3 p.m.?
Yeah.
Do all of you drink coffee before? Why doesn't anyone? you were smart. You have coffee. I see coffee. I feel like I should require everyone to bring coffee. Or wait, someone suggested to me that we do jumping jacks midway through this class. You know that makes me want to do it more, right? We won't do jumping jacks. But if I see anyone sleeping, we're doing burpees. Okay. All right. So repair mechanisms brings us down to 1 in 1 million base pairs. That's not very much change. Inherent meiosis, right? What I talked about last time are all these other events, though, that happen on the reg. Oh, sorry, that seems weird to me. That happen regularly. I don't want in your notes to be like on the reg. Right? So crossover, that happens every time during meiosis. Crossover occurs, right? There is equal and unequal crossover events that happen pretty regularly during meiosis. So those can lead to pretty significant changes in a single meiotic division. So really, evolution is this kind of iterative process of having these events that do lead to highly significant changes, or can lead to highly significant changes in the genome, and then the slow process of acquiring mutations that that may or may not make a difference, right? This is why evolution takes thousands and thousands and thousands of years, right? Have you guys seen the? I'm not going to talk about that. I've already gone on enough tangents. Okay. Have you guys seen the Jesus Saves guy?
Oh yeah.
I went to get coffee today, and I sat down initially with my back to him, but then I was like, "What's happening over here?" It's like, "Oh, this is like watching TV." It was really interesting. I mean, hey man, give it up for his passion, right? But it was so interesting because there were all these students challenging him on the molecular basis of evolution, and but his response, there was a student next to me who was like, "I'm just trying to study." I'm like, "No, this is interesting. Like, I teach this stuff. He's getting it totally wrong. Like, he needs my class." The student was like, "Oh my gosh, why did I talk to her?" Yeah, it's fascinating. So if you guys want, I mean, that might be a good study technique, not to do it in a condescending way, but just to, I mean, go out and try to spread some knowledge. Oh, okay. Yeah, I mean, hey man, to each his own about their beliefs, but I always find it interesting when your beliefs prevent you from understanding things that there is just evidence, like mounds and mounds of evidence for, right? That's interesting. So anyways, okay. All right, bring back. I have in-class activities. I feel like I'm not going to get to them because of conversations, but maybe we need these because it's all right, so another way to get genetic changes is something called reactive oxygen species. And so when they were talking about the damages from Hurricane Harvey, I heard that there was a chemical plant that exploded, right? They were creating oxides, and so instantly I thought, oh my gosh, I hope there aren't people anywhere nearby there, because reactive oxygen species can cause really significant damages to DNA. And so reactive oxygen species are essentially things like superoxides, peroxides. They are oxygen species that have an unpaired electron in their outer electron shell. Yeah, the biochemistry students like shell. Yeah, outer electron shell. And so that means that they're really unstable and reactive, right? And so what they do is wreak havoc within the cell if there's too many of them. Our cells have normal antioxidant mechanisms, but there needs to be balance achieved, right? So if there are too many reactive oxidant species for our antioxidant mechanisms to address, what happens is those reactive oxygen species react with things like proteins and DNA and cause them to break down. And so, but normal metabolic processes produce reactive oxygen species, which is part of the reason why we have these built-in cellular mechanisms to try to address them before they do wreak havoc in our cells. But we're also very good at overloading our bodies, right? So for the people who tan and chain smoke, right? So those are two of the big ones. Cigarette smoke UV rays, X-rays, that's a lot of times why, doctors wanna be sure an X-ray is necessary before they do it, because that can cause damage, and that's why they always put the, the lead barrier to protect those gonads, right? So you don't create mutations in your germline that get passed on. That's the reason. Right, so all of these things can cause all sorts of changes in DNA. Reactive oxygen species have been associated with gene shuffling. Yeah.
Is ROS a synonym for free radicals, or free radicals a result of ROS?
Free radicals aren't they a type of reactive oxygen species? Because they're oxygen radicals. I think they're a subcategory. That's a good question for Dr. Google. I'm gonna ask them. Yeah. So I know a lot about reactive oxygen species, and I think they are a type of oxygen specific free radical in our cells. I could be wrong though. Ask Dr. Google. Right, so they can alter the way our genes are regulated, which is really important, right? Because genes aren't supposed to be expressed all the time. We have certain genes that need to be expressed at certain levels and certain cell types, and so when that's altered, that can absolutely lead to all sorts of disorders, cancer being one of them, right? Because if you start expressing, for example, a gene that promotes proliferation in a cell that's not supposed to be proliferating, that's step one in moving towards tumors. Uh-oh. Someone's gonna cause you guys to burpees. Just kidding.
So the ROS, depending on whether they call it for mutation, it could be germline or the subtype.
Yeah, that's right. So reactive oxygen species can affect somatic cells or germline cells. Yeah, absolutely. Reactive oxygen species, it's just what cells they're affecting. And again, right, we have all these sweet DNA repair mechanisms, thank goodness, that can try to reverse mutations or genetic changes, but again, the cell's not 100% efficient. So the repair mechanisms are not 100% efficient, which is why just say no to smoking. Yeah.
I might have misunderstood one of the other slides. I thought that you said environmental changes couldn't cause.
Directly cause mutations, right?
Yeah.
So that's a really good point. So I think really what's happening, and I'm gonna have to address or make sure I understand this, but I think it correctly. But I think what's happening is kind of the same thing, right, with UV irradiation. The way that it damages the DNA then affects the way that the DNA, right, is regulated. Does that make sense? So it's not directly leading to the mutation, but because it's causing a structural change, it then, yeah. Okay. Good questions. All right. Transposable elements. So fantastic. So transposable elements are mobile DNA sequences that can colonize genomes and move around within them. So let's talk you through this picture right here first to help explain it. So in this image, this is supposed to be just a strand of DNA. And the orange and yellow is the transposon. So right, it's hanging out here, and then it becomes mobilized and inserts itself into this other region of, in this case, the same chromosome, and inserts itself right into the middle of the purple gene, right? And so if this happens, if it inserts itself right into the middle of another gene, it's going to completely alter that gene, whether it's in the genetic regulatory element or in the open reading frame. That's going to affect the way that gene is either expressed or, if it's the open reading frame, the amino acid sequence, right? Yeah. This would be regarded as an insertion, and so the thank you for saying that. So a lot of times, and this is getting a little semanticsy, but when people talk about in molecular cell biology mutations, oftentimes they're referring to like a discrete change, so like a single base pair change, right? Which is why on that slide it was just showing one base pair changing, and like in a frame shift, a single base pair being inserted or removed. But this is also a type of genetic change that's referred to. There are large insertions and deletions, and I think when people are being a little bit more relaxed with their terminology, which is what happens when you're outside of a classroom, these are also regarded as general types of mutations, but they're not point mutations, right? So this would be something called an insertion. And then what transposons can do, right, because they're like nomadic DNA, maybe they decide that they want to leave here too, right? So it's hanging out in the middle of purple gene, and it seems like, oh, well, if it just left, purple gene could join up again and be happy purple gene and encode its happy purple protein, right? But it turns out that transposons don't always have what's called a perfect excision, right, excising themselves. Often what happens is they have an imperfect excision. So they hop out of the chromosome and they're like, hey, purple gene, we like you. We're going to take a little bit of you with it, whatever. So it removes part of the purple gene sequence so that when it, if it religates, it's also changed the open reading frame of that gene. This is actually a mechanism that's used in laboratories to intentionally create mutations. So scientists will intentionally insert transposons or remove them to try to create changes in the genome of cells for animal models. Okay, so these were first identified in the mid-40s by Barbara McClintock. She was a badass scientist, right, in the 40s when there were not many female scientists for sure back then. And she identified this in corn, right? She was studying maize. And mid-40s, she reported it, and no one in the scientific community believed her because it went against conventional wisdom, right? And so her paper on it wasn't even published until 1953, and people still in the community kind of poo-pooed it. Along the way, she also noticed, like, hey, not all genes are expressed all the time. Like, they're expressed differentially during development and in different cells. And people were like, they just didn't pay attention. It wasn't until the 70s when some male scientists found the same thing that they went, oh, that lady scientist way back in the 40s, she was right. And so then she was given the Nobel Prize in like 1983. So like 40 years after she made this discovery. I mean, it was 30 years before people realized that it was inauthentic and, I mean, really revolutionized the field in the way that we think about DNA and gene expression. Amazing. Okay, so transposons, right? Again, they're also called transposable elements or P elements. They can disrupt gene function, alter regulation. Right, you can create, in theory, create a new gene if this gene inserts, integrates in with the host gene segments. Over half of all human DNA has homology to known transposons. So that other, 92% of our genome that we're going, what is this DNA sequence for? A lot of it has homology to transposons, which have homology to a lot of viral DNA. So I was talking to Dr. Holland, who teaches evolution here, about it, and he was saying that that's one of kind of the prominent hypotheses in the field of evolution, is that it's just kind of this ancient DNA that we acquired from viruses. It's not actively jumping around, but there wasn't any selective pressure to get rid of it. So it's just still there in our genome. So it remains to be seen if that is in fact the case, but it's interesting. And it doesn't seem like we have active transposons in our genome right now, but a lot of other organisms do. So in mice, for example, there are transposons hopping around their genome and driving a lot of evolutionary change in those organisms. All right. You guys good?
335.
All right. Yeah, question.
You said it can potentially make a functional change even if it inserted theoretically.
In theory, yeah. I don't know of an example of that happening, but in theory, yeah. the only thing that I've ever really read about is more, historically how it kind of moved genes around in corn, right? I learned about its discovery, and then how it's used in a laboratory setting to manipulate genes. So I used this method, a gene that I was studying as a graduate student, I wanted to get rid of the gene and see what the phenotype would be, right, in the organism. So I did a genomic analysis and saw, oh, there's a key element right next to this gene. So what I did is I introduced an enzyme that would cause that transposon to be excised, and then I did that in like hundreds of different fruit flies, and then I went through one by one and looked for ones that had an imprecise excision that removed expression of my gene. Right? So it took, in this case, like imagine the purple gene was my gene of interest. It took some of that with it, and when it took that, the gene was no longer expressed. But in theory, it could create a hybrid gene. I don't know an example of that though. All right, so we're gonna switch gears now. Yes.
Sorry, like interest level is bigger than the sequence and have the ability to potentially
Mobilize. Yeah. Usually, so the reason why, this is more detailed than you need to know about transposons, but since you asked, the reason they colored this yellow, the way that we can recognize them when we do a genomic analysis is that the ends of transposons have really stereotyped sequences flanking the transposon sequence. That's why they're yellow. So, right, you can do a genomic analysis and software can just basically analyze where they see those sequences. Those are identified by a type of exonuclease, a very specific type of exonuclease that basically cleaves at those sites in theory. Is that what?
I got it.
Okay. But just sometimes it will bind to the yellow, but then cleave out here instead of cleaving at the yellow. Does that make sense? Yes. Okay.
So it always gets it wrong.
Not always. Sometimes it has precise.
So
Sometimes
does it always leave extra, or does it always take from the
Just surrounding
DNA?
it?
It never leaves itself, and does it always leave, or does it still like hang around? Sorry, you don't have to answer it if it's too. I was just curious.
Oh no, I have to answer. And I will always, so just so you guys know, I will always try to give you the best answer I can. And a lot of times it's, I'm deducing things from what I do know, and I'll always be really clear about when I'm doing that, right?
Yeah.
If I don't know an answer, that's when I'm like, I don't know, this is what it could be, and I'll look it up and get back to you. Because it turns out no one knows everything. If anyone tells you they do, and they're scientists, call BS on that. End the conversation, because they're not a good person to talk to. Okay, so sometimes they have precise excisions, sometimes they have imprecise excisions. I'm not totally sure what governs whether it's precise or not. Yeah, and then does it ever leave itself? Not that I'm aware of. But yeah, it's totally possible. And then how do they become inactive? I'm not totally sure. I mean, my educated guess is that our cells stopped expressing the particular exonucleases that recognize the sequences. You're welcome.
Real quick, in this picture, the transposon used to be in that lower corner.
Oh, I know what you're gonna ask. Did it leave, or can it replicate itself and then be inserted there? Yes, to both. Both can happen. So I guess that address. So okay, that gets back to your question. That gets back to your question too. So sometimes it excises and inserts somewhere else, but there are mechanisms that they found where it replicates itself, and then that replicated piece inserts somewhere else. Exactly. Exactly. Future professor over there. All right. So this is a review on how we go from nuclear RNA, sometimes referred to as pre-messenger RNA, to a functional protein, right? I'm gonna change up sides to change it up so I don't have my back to everyone over there. So, all right. So, right, the example is with beta globin. so we start with the gene, so this is supposed to be the gene hanging out in the chromosome, right? The process of transcription causes the production of nuclear RNA. Nuclear RNA has to be processed. So some of the processing that we're going to talk about in lecture 6-7 are the addition of the 5' cap and the addition of the poly A tail. So it gets these additions on either end that help protect it and influence export and splicing and translation, all this super fun stuff. And then the introns have to be removed, right? And so when it's fully processed, then it's regarded as mature messenger RNA. That's what gets to leave the nucleus and get translated always in the cytosol into a peptide. That peptide, right, when you're not done, the peptide has all sorts of post-translational modifications. So post-after translation, post-translational modifications that occur and help influence its folding. So it can get glycosylated, big old sugar groups get added and modified, lipids can be added, all sorts of fun stuff. And then the protein folds. If sometimes peptides lead to just a functional monomeric protein, so single peptide. Sometimes you have proteins that are part of a oligomeric protein. So in the case of hemoglobin, you have 4 subunits, so it's referred to as a tetramer. Right? If you have 2 peptides, dimer, 3 trimer, 4 tetramer, so on and so forth. Okay. Did I hit that all? All right. So I know this is all review. I'm saying it because it is very important that you know this like the back of your hand. I was actually talking to a professor once, and I'm not going to say who, but they do not teach molecular, and they're like, oh yeah, I still get transcription and translation messed up. What are you saying? No. Okay. So genetic code, right? There are, it turns out, 22 proteinogenic amino acids, 21 used by eukaryotes. I know that a lot of you are like, no, there's only 20. There are 20 that are kind of normally encoded by codons, and then there's one that has like this special modification, I think selenocysteine. So that's just a fun fact. I'm never going to ask you about it. But maybe if you're on like biology or Jeopardy, that would come in handy. But just FYI, there are a few extras. So there's 64 codons, 61 code for amino acids, 3 encode stop signals, and the genetic code has redundancy. We already touched on that. That's the whole wobble-based thing. So it just turns out that the tRNA molecule at the anticodon site, the first 2 positions have to be paired up perfectly. The third, not so much. And so you can see here, right, for leucine, for example, you need CU in the first 2 positions, but the third one can be whichever, right, U, C, A, or G. In some cases, right, there's wobble, but it is a little bit more stringent. So here, right, AA can encode, the first 2 positions can encode asparagine or lysine. But if it's a U or C at the end, it's asparagine. If it's A or G at the end, it's lysine. Okay, so wobble can vary. The take home is genetic code has redundancy because of that wobble position. All right, so even though multiple codons can encode one amino acid, the reverse of that is not true. There's no one codon that will ever encode more than one amino acid, right? So AUU is always going to be isoleucine no matter what. And the genetic code is nearly universal. That's pretty fantastic, right? So a CUU in us is leucine. A CUU in bacteria is leucine. A CUU in My Little Pony, if they were real, would be leucine, right? So it's, I mean, that's pretty fantastic, in my opinion, right? Big happy family, man. Or I wish we were all happy. Okay. you're not all happy. There are a few exceptions, right? And that is just like in a few organisms, there are a few codons that are different, right? So Drosophila, I think like AGA or something, encodes something different than arginine. So there's a few random differences, but by and large, it is universal. It has to be read in frame. So this is just revisiting of the idea of how significant frame shift mutations, how significant the damaging they can be. Right, so usually the start codon, that dictates the frame that's read. So the ribosome, through mechanisms we will talk about, recognizes that start codon and then starts just reading in a pattern of 3, right? But if a gene has a frame shift mutation, it shifts everything downstream of it. So there's always going to be 3 potential reading frames because, right, the DNA code is read and codons that are composed of 3 nucleotides. So right here, if before when I was talking about frame shifts, really what she's saying, here it is kind of spelled out in a little bit more detail. So if the gene is supposed to be read CUC, AGC, GUU, okay, that codes leucine, serine, valine. The next potential reading frame, I don't like the way they did this. Wait, I don't like the way they did this. Oh no, it's okay. So if you're starting reading from there at CUC, if you skip that C, right? If you skip the C, instead you're reading UCA GCG. So serine, by happenstance, is the same, but then next it's changed. All those amino acids, right? Now you have alanine, leucine, proline, which is totally different. So if you skip the C, so this is why I don't.
I was gonna skip the C and the U.
Yeah, skip the C and the, okay. Dude, okay, apparently, it's funny, Chloe, my TA before, she's like, hey, do you want anything from Starks? And I was like, oh, no, I'm good, I don't need coffee, like, wrong. So skipping the CU, right, now you're starting at the C, reading CAG, right? So CAG, CAG, CGU, and if you're confused, even people with PhDs apparently are confused by looking at this chart, given this lecture like 5 times before. So right, there's 3 potential reading frames, and if that gets shifted, it's changing all the amino acids downstream of that frame shift mutation. Do you need an assist? Happy to oblige. All right, so key players in translation, right? mRNA, tRNA, ribosomes, amino acids. These are all your friends that you should have become familiar with a long time ago. If you're confused on what these are doing, come talk to me. I'm not going to spend too much time, or really any time talking about it right now, because I think we should all be on board with these molecules. So this is just a reminder of, if you don't know what these words are, come see me in office hours, and I'll talk at you about them for a long time. One protein you may not be aware of that is critical for the process of translation are a protein family called eukaryotic initiation factors. These are proteins that we're going to talk a lot about in lecture 6-7, and they're really critical for bringing a lot of these pieces together. So they help with the mRNA, basically getting the initiation complex together, recruiting the ribosome, recruiting the first tRNA. So we'll talk about those more extensively. All right. So now protein structural organization. Have you guys learned about primary, secondary, tertiary, quaternary structure before?
Yeah.
Oh, fantastic. Okay, well this will be fast too then. So right, primary structure is just like reading words of the sentence. It's just the amino acid sequence. Secondary structure, right, is this repetitive folding that's caused by hydrogen bonding in the repeating units of the peptide backbone, right? So by repeating units, every amino acid has an amine group and a carboxyl group, right? You also have the R, the side chain. the amine and the carboxyl group. So the amine is always gonna have a hydrogen coming off of it, right? Carboxyl group is always gonna have that double bonded oxygen. Well, what happens is the hydrogen of the amine group forms hydrogen bonds with the oxygen from the carboxyl groups, right? All across the protein. And so the two structures that are caused by that repetitive hydrogen bonding is something called an alpha helix or a beta sheet. And so the alpha helix is also sometimes called a coiled coil, so it forms kind of this spirally structure. It's reinforced by these hydrogen bonds. So everywhere you see the dot, 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 that's the hydrogen bond. So oftentimes proteins have both alpha helices and beta sheets. And it just depends on what protein you're talking about, how many alpha helices and how many beta sheets you see. Alpha helices are always drawn as a spiral. Beta sheets are usually drawn as arrows going in different directions. So I'll show another example of that in a few slides. All right, so any protein you look at is probably going to have this, either a number of alpha helices and or beta sheets. So that's secondary structure. Tertiary structure is the full three-dimensional folding that is also influenced by those side chains. And so when a protein folds, both of these things are happening at once. Both secondary structure and tertiary structure are occurring. It's just one is caused by the hydrogen bonding of the repetitive units in the peptide backbone, and the other one is more influenced by those R groups. R group dance. And yeah, absolutely. Even I was predicting a question when I said, yeah, absolutely. The question that I was predicting, some might be going through some of your heads, is why would a protein form either alpha helices or a beta sheet? Do R groups influence whether alpha helices or beta sheets form? And the answer is yes. So those R groups are pushing the protein to form its most optimal conformation. And the way the R groups are pushing and pulling can influence whether those hydrogen bonds create an alpha helix or a beta sheet. They're not directly involved in the bonding itself, right, the bonds that form, but they can push the structure of the protein to favor the formation of alpha helices or beta sheets. All right, and the final structure, quaternary structure, that only exists if you have a protein that's composed of more than one peptide, and quaternary structure refers to that final conformation that occurs when all the peptides find each other and form their final functional structure, right? So in the case of hemoglobin, right, every textbook's favorite example, you have two beta globins and two alpha globins that hold like that, form an extra. All right, so now there are slides dedicated to each of these, so it gives you an opportunity to shout out any questions if any of that is confusing to you. All right, again, primary structure is just the sequence of amino acids. It doesn't indicate anything about folding, it's just, right, glycine, proline, threonine, glycine, it's just that sequence of amino acids. Right, an amino acid, peptides always have an amino end and a carboxyl end. So DNA, right, so RNA is transcribed 5' to 3'. The 5' side is what is created first, right? The 5' side is what is translated into the amino end, right? So the amino end of a peptide is always created first. So primary structure, amino acid 1, is going to refer to the amino acid that is at the amino terminus of the peptide. Okay, is that review too? No? Yes? Okay. It's been a whole summer probably, at least, since you guys have heard this stuff, so I'm a fan of review. It's good to go, oh yeah, I learned that, but I need to learn it again. Secondary structure, again, right? It's the hydrogen bonding between those repeating constituents. In every amino acid, you're gonna have a carboxyl group and that amine group, and the oxygen from the carboxyl group forms hydrogen bonds with a hydrogen from the amine group. Okay, and so here's the cartoony way of depicting a beta pleated sheet, the arrows going back and forth, and here's the cartoony way of showing that alpha helix or coil. Right, and so just again, overall conformation of each protein is going to be unique. And these are the most common folding patterns seen within a protein, and a single protein can totally have both, they often do. Finally, tertiary structure, and you name the type of interaction and it can exist, right? So it's just for tertiary structure, it specifically means what interactions are happening between those side groups. And yeah, hydrogen bonds can happen between those as well. We can have disulfide bridges, which is a special kind of bond that forms between what amino acid? Anyone? Cysteine, yeah, it's a sulfur-containing amino acid. You can have ionic bonds, van der Waals forces, or van der Waals interactions, hydrophobic interactions. So those are all interactions that are going to influence the way that protein folds. Because at the end of the day, it wants to form the most stable conformation. And another consideration proteins have when they're folding is, are part of them going to be in the membrane? Because the membrane that's composed of lipids, right? Are lipids hydrophobic or hydrophilic? Hydrophobic. All right, think about throwing oil into water when you're making spaghetti. All the oil's like, and aggregates, right? That's what amino acids, if there's a hydrophobic side chain, it doesn't want to be exposed to lipids. Hydrophilic side chain doesn't want to be exposed to lipids, hydrophobic ones do. So what you'll see is if you have a protein spanning the membrane, the part that's spanning the membrane, you'll see hydrophobic residues kind of facing the outside. Whereas you would expect to see hydrophilic residues hidden. But if you have a protein that's folding like in the cytosol is mostly water, it's aqueous. So hydrophobic side chains are going to want to internalize themselves into the protein. And so proteins try to maximize how many hydrophobic moieties are internal and maximize hydrophilic residues on the outside. I talk more about that in a later lecture as well. Yeah.
So secondary structures, they're folded, but they're not like, they don't have the disulfide, they're not like the final.
Like you guys asked questions. So the secondary, it's not like proteins go through, like I'm primary, now secondary is happening, now tertiary is happening, it's all happening at once. So proteins are folding, all these interactions are happening at once, right? Because they're just molecules. They're not going to be like, oh, let's let all the carboxyl groups and amine groups form their hydrophobic residues first, right? That's not how it works. They're just trying to form the most energetically favorable conformation wherever they're folding, whether it's within the context of a membrane or within the cytosol or lumen of the ER, which is where those things happen. And so all those things kind of happen together and can influence each other. Does that make sense? Kind of, for 4 p.m.? Yeah, so it's not like the 1, 2, the 2 and 3 don't happen, it's 2 and 3, they kind of happen together. And those are just terms given to that type of structure. So if someone says, refers to the secondary structure of a protein, they're referring to the alpha helices, the beta sheets that are caused by that hydrogen bonding from the amine carboxyls. If they refer to tertiary structure, it's more the global structure that also includes all the R group interactions.
Ooh, so this even happens before translation is finished, right?
Yeah, it usually starts happening before translation finishes. However, that's a much longer answer that involves discussion of chaperone proteins, which is in a later lecture.
Okay.
To be continued. Or office hour.
Yeah.
Yeah. Alright. Cool. Alright. It's always really hard for me to be like, it's coming next. I super want to be like, and there's all these different chaperones, and this is what they do, and then, yeah. I get off track and half the class is like, what is she talking about? Shut up. Why'd you ask that? So quaternary structure, again, a lot of proteins don't just function as monomers, they function as larger oligomeric complexes, right? So collagen is one example where you have multiple peptides that form this kind of fibrous braided structure. And then the fibrous braided structure cross-links with other fibrous braided structures. It's pretty cool. Or hemoglobin, right, which you all know well by now functions as a tetramer. And so the quaternary structure refers to that final functional structure when all the peptides are able to fold together. Protein coming, everyone. Protein coming. Okay, sorry. So it's really difficult to predict final protein structure from its primary sequence, right? From the primary structure. The primary structure is just the amino acid sequence. So most proteins actually go through several states on their way to their final stable structure. So it's kind of this iterative process of, ooh, does this feel right? Not exactly. Let's move this over here. It's like you know, when you're in bed trying to get comfortable. It's kind of what proteins are doing. They're trying to get comfortable in their environment. There are software programs that are getting better and better at making predictions of what a final protein conformation is going to look like. But there's this whole field called protein crystallography. Has anyone heard of protein crystallography before? Okay, yeah, the biochemist has. Any biochemist has. So protein crystallography is a type of science where they basically try to take proteins and crystallize them, and then essentially it's really hard because you try to get the proteins to fold into their native conformation, and then essentially measure what the structure looks like and make these kind of computer renderings of the structure. It just seems incredibly boring to me. I had a friend who did protein crystallography in graduate school, and she just had these glass trays, and in every well she's changing like a subtly different pH, or changing it to a subtly different pH or a subtly different protein concentration. It can take years and years and years to get an efficiently crystallized protein. It's extremely challenging. And so when protein crystallographers finally get their crystal structures, right, a computer will generate this cartoony image of what the structure looks like. And that's what you're looking at right here. This is the protein structure of a protein. You can see here that it has all these arrows are representing beta sheets. All the coils are representing, right, this coils, the alpha helices. And so what I want to talk about now is kind of something a little bit different than the primary, secondary, tertiary, quaternary structure. I'm going to talk about something called a protein domain. So protein domains are substructures within the protein. What I mean by that is here we have the one, this is the big old protein structure, right? It's a single peptide, so this is the tertiary structure of this protein, right? Here's the amino terminus and the C terminus. And these domains, or these regions, what they realize is each of these regions has their own specific function associated with it. And, moreover, they can fold independently of the rest of the protein. So if you were to chop off the green, yellow, and orange domains, the blue region would still form this same kind of discrete structure. So when you have a region like that has the ability to kind of fold independently, and that's associated with a specific function, the name that's given in the field is a protein domain. There are all types of different protein domains. So I talked about FMR1 the other day, right? And I mentioned that FMR1 encodes a protein that binds to mRNA and inhibits its translation. Well, FMR1 is a big old protein. There's only one part of it that binds directly to the RNA. That region will be called the RNA binding domain. Transcription factors all have DNA binding domains, right? Because they have to bind DNA and influence transcription. You can have protein-protein binding domains, where you have a protein that binds directly to another protein. All right, so more details. Right, different domains are associated with specific functions. Okay, that's just a domain fun fact. Usually they're between 40 and 350 amino acids, but there are domains that are as small as like 12 amino acids. I like that I showed you a 3 and said 12 amino acids. My kids are doing great at counting. So what you're looking at right here is a protein that's referred to as SARC. It has, let's see, it's a protein kinase, and the kinase would, oh, right, so the kinase region is over here. And so the SH2 domain and SH3 domains, those perform regulatory functions, right? So maybe they bind to specific proteins that influence enzymatic activity, but they're not actually the part that, oh, do you guys remember what a kinase does to other proteins? Phosphorylates them, right? So it adds a phosphate group to their target protein. So the region that's responsible for the kinase activity of the enzyme for transferring the phosphate are the yellow and orange lobes, right? So those would be referred to as the kinase domains. And the other cool thing about these domains is that these are the domains that often determine belongingness to certain gene families, right? So remember way back last Wednesday, I was talking about gene families, and I gave an example of expansion of gene families, and I talked about basic helix, loop helix, transcription factors. Basic helix, loop helix transcription factors have really similar looking DNA binding domains, right? So that's kind of one of the things that determines belongingness to that gene family. I told you the story of my dissertation project where I studied a gene that no one is, I told you guys that, right? I studied this gene that hadn't been studied before. I just had the sequence. And when I did some bioinformatics analysis of the sequence, what came back was it had this 12 amino acid motif called the lipocalin signature motif. Usually that motif was enough for proteins to be regarded as lipocalin family members. So that gave me a hint as to what its function was and to what gene family it might be a part of. So, right, because the protein domains are responsible for function, that means those are going to be the more conserved parts of those proteins. Right? Yeah.
Does the folding have anything to do with the function of the protein domain?
Of the
Protein domain?
yeah, well, yeah. I mean, the folding is right. Folding is everything for a protein, and so the ultimate tertiary folding of a protein is going to lead to functional protein domains. I feel like I'm not answering your question based on your hard stare.
Like in the first paragraph where you say we could fold independently.
Yes. Yeah. So when the proteins fold, it's like a folding party. They're folding, but it turns out that the way that the kinase domain folds, it's kind of happening independently of what's happening over here. So you could, in theory, change the peptide sequence of that leads to the green region and alter its ability to fold, and the kinase domain could still be intact. Right? So it would alter maybe regulatory functions, but it wouldn't alter its ability to function as a kinase. Does that make sense?
Yeah.
Okay. Yeah.
Is this one protein or is it like
This is one protein. This is one protein with multiple distinct domains, right? The kinase domain, the SH3 domain, and the SH2 domain.
If it was like hemoglobin, they wouldn't be domains since they're four different.
That's right, that's a different thing. So in the hemoglobin, you have different peptide subunits. But each of those subunits has an oxygen binding domain right? The heme group. So, but each of the globins have that same oxygen binding domain. Yeah, or heme binding. Yeah. Okay. So we have a few minutes. We have 5 minutes to work on my in-class activity that I'm going to change because it's, well, whatever. I'll just leave the questions up and you can do with them what you will. Okay. Exit. Did I tell you? All right. All right. Since we only have 5 minutes, what I want to do is let's look at question 1 right now. All right. So you're studying a gene. I just picked a gene, any gene, neuroligin 1, and notice that it's been duplicated, but the sequence of the second neuroligin 1 gene is identical. Would these 2 genes be considered a gene family, and why or why not? Let's just have a, okay, no, that is the correct answer. Would someone like to raise their hand and explain why? Everyone's whispering, ah! I have a hand over here. Exactly, what I said at the beginning of class. This was my test. Was everyone paying attention to me at the beginning of class? Right, because what's the definition of a gene family? Are they identical genes?
No.
They're subtly different. They're similar, right? So, would you expect these to have similar functions or identical functions? Okay. I feel like I'm like a 2nd grade teacher right now. Like, all response, everyone. Okay. So this gene encodes a single peptide that forms a monomeric protein. If hydrogen bonding is prevented, what levels of protein would you expect to be disrupted? Secondary for sure. Would tertiary be affected?
Oh, okay.
Yeah, right. Again, think about folding. So that's why I put this question up here, right? Because always when we see these lists and when you attribute like primary, secondary, tertiary, it seems like successive. But if you mess up any folding, it's going to disrupt all the folding, right? So you're for sure you're directly screwing with secondary folding. Hydrogen bonding can also happen between R groups, so it's going to affect any tertiary folding there. But if you mess up the ability of hydrogen bonds to form, it's going to affect the way that the R groups are positioned next to each other too. Mess up any folding, you mess up all the folding. Yeah? Okay. Except, right, if you're talking protein domains, which can kind of do their own thing irrespective of other domains. Okay, finally, about 20 amino acids of this, the protein neuroligin 1 encodes binds directly to another protein. This activity is critical for its function. These 20 amino acids can fold on its own into a discrete substructure. What's that region called? Protein domain. And it would be even further called a protein binding domain. Protein.
Cool.